urgent message from him. He would like to send his apology to you and everyone because he will be join a little bit later than expectation. I think around 8:30 because he's currently on his way back from a work trip in Đồng Tháp province.
Okay.
But is currently experiencing heavy traffic.
Okay. So later on you will introduce me first before I start. Is that correct?
Yeah, I will be hosting on behalf of Mr. Lai until he joins. I don't know, but maybe 8:30.
Okay. All right. Thank you.
But I think we should wait a little bit until
Sure.
7 p.m. I think.
Okay. Just want to test out if my mic is working well. Can we hear me well, right?
Yeah.
Okay. All right.
Okay, thank you. Chị Tuệ, chị có vô không chị Tuệ?
Có Thảo ơi.
Hay lại đi gì từ Đồng Tháp về mà bị kẹt xe 8:30 thì mới vô á chị.
Không.
Nên là em hot say nhưng mà cái cancer em không có biết nhiều nên có gì chị Tuệ hỗ trợ em với nha.
Ok em.
Cảm ơn chị. Chắc 7 giờ em nói lại cái vụ thầy đi trễ giúp cho mọi người biết luôn rồi cái gì mình đã bắt đầu rồi chị, tại giờ chắc mọi người cũng vô chưa có đủ.
Ok không sao, cứ có thể trễ hơn khoảng 5 phút cũng được, tại vì chị thấy ở đây thì hình như chắc mới có 15 người thôi.
Mèo. Mày có chừng á.
Hello, Ray. How are you?
Hello. Hi, Dr. Lui. Hello.
Hello.
I heard that you are stuck in traffic, but you managed to.
Yes. I'm on the bus now, on the way back to Ho Chi Minh City. Yeah.
Okay. But you managed to still dial in.
Yes. But
Okay.
I have Miss Thảo will help me. Yeah.
Okay.
So no worry.
No worries.
Ừ.
Chắc mình chờ tới 7:05 đi xíu thôi thầy.
Chắc mình chờ tí xíu. Em cứ tiếp tục dẫn nha Thảo ha. Đó. Tại tôi sợ giống như một lát đó tôi không biết là trên đường internet nó có tốt hay không. Đó. Đang ở từ Đồng Tháp về mọi người. Right? Can you please share the screen?
Sure.
Yeah, just to make sure that the sharing it work well. Yeah.
Sure. Can you see my screen properly?
Yes. I can see your screen well. Yeah, thank you.
Can you move to the next slide to make sure? Okay.
Yes.
Okay.
Yes. On good. Yeah, thanks a lot.
Thank you.
Okay.
Ở bên Tuệ An á em Tuệ Minh Tuệ. Đúng rồi Minh Tuệ. Ở bên của em có ai nói nữa không? Thì Minh Tuệ ơi.
Chị Tuệ ơi, hình như có mình chị Tuệ là thầy mà.
Dạ có mình em thôi anh Lợi ơi. Chỉ có Ray với em thôi. Dạ. Nhưng mà chắc chỉ có Ray nói thôi. Dạ.
Ok. Em không nói luôn cái gì luôn hả?
Dạ không, em hỗ trợ thôi.
Ok.
Dạ.
Vậy có gì lát Thảo giới thiệu Minh Tuệ
Dạ
luôn
có,
ha.
dạ thầy.
Ok. Tại mình cứ chờ tí xíu á, tại thường mấy bạn cũng phải phút thứ 5, thứ 10 mới vào. Cái văn hóa khó mà thay đổi được đó mọi người. Đó. Bởi khổ lắm.
Hi Ray, we are saying that we will wait until 7:05 and after that before we start, please allow me to say a few words in Vietnamese to our participants, okay?
Okay. Sure.
Okay, thank you.
Mọi người chịu khó chờ tí xíu ha. Chờ thêm khoảng 2-3 phút nữa. Đó, để mình bắt đầu cùng một lúc luôn nha mọi người. Cảm ơn mọi người.
Dạ vâng thầy.
Dạ em chào mọi người, chắc là mình bắt đầu luôn ha. Thì tối nay á, như mọi người cũng biết thì mình sẽ à, tìm hiểu về ờ, ưu điểm của công nghệ HiFi PacBio trong cái việc mà Somatic variant detection thì nó là long read. Thì long read thì như mọi người cũng đã biết về PacBio rồi, thì nó có cái độ dài và cái độ chính xác khá là cao, nên là sẽ có những cái ưu điểm khi mình giải mã những cái vùng gen khó, những vùng challenge rồi, hay những cái biến dị cấu trúc lớn. Nhưng mà nó cũng có một cái nhược điểm là nó có cái độ chính xác là không có bằng Illumina. Nhưng mà sau này thì PacBio họ có thêm cái công nghệ onso nữa, thì nó cũng giải quyết luôn cái vấn đề đó, tức là nó vừa dài và vừa chính xác. Thì độ chính xác của onso như thế nào, cũng như là
Recording in progress.
em quên record. Thì cả cái long read HiFi và cái also này nó sẽ giải quyết những cái vùng challenge trong cái somatic variant detection như thế nào. Thì mình sẽ cùng tìm hiểu qua cái sự trình bày từ cái nhà cung cấp PacBio, từ cái đại diện từ phía PacBio là anh Ray. Anh Ray là chức vụ là Territory Account Manager của PacBio. Ngoài ra thì mình còn có cái sự hỗ trợ từ chị Tuệ là từ phía DKSH, là nhà cung cấp của PacBio ở Việt Nam. Thì tại vì cái kiến thức về cancer của em nó cũng hạn chế, nên là mọi người, nếu mà anh chị nào có thể đặt câu hỏi trực tiếp tới anh Ray luôn thì mình đặt câu hỏi, hoặc là mình cố gắng mình type cái câu hỏi của mình vào chat box mà nó rõ một xíu để em đọc lên cho anh Ray, anh trả lời. Mà tốt nhất là mình hỏi trực tiếp luôn thì nó sẽ dễ hơn xíu. Dạ em cảm ơn mọi người. Hi Ray,
Yep.
can you hear me? You can start your presentation now. Can you share your screen?
Okay. Can you see my screen?
Yes.
Okay. All right. Okay, I will start. Okay. Thank you so much, everyone, for spending Sunday night with me to actually, go through a presentation about how, PacBio is changing the paradigm for cancer genomics, with our solution of both accurate long and short read sequencing. All right. So I think an interesting about PacBio is like we are the only company right now with both the long read as well as the short reads technology. So for the long read, we actually have our HiFi sequencing, which actually delivered our long reads with the highest accuracy, to actually, provide comprehensive characterization of the genome, not only the genome, but, in the multiome itself, genome, transcriptome, as well as epigenome, which I will talk more later on. And then we also have the short read sequencing platform called our ONSO system as well, which is based on the sequencing by binding technology. So it's for the SBB sequencing, it's 10 to 100 times more accurate than the conventional NGS approaches. So with that, right, we can actually cover a lot of different applications for cancer genomics, as you all can see on the screen here. And besides the different application as well, we can also support multiple different sample types, depending on, what you want to look at. So we can support, fresh frozen tissue, blood, FFP, and liquid biopsy on the short read side. And later on as well, I will share a little bit more about the insight or the advancement that we have for long read for FFP as well, which is very exciting. Okay, so the first section, I will talk more about HiFi sequencing, which is our long read sequencing. So how do HiFi actually works, right? It's actually, we usually extract high molecular weight DNA. And then we will start with this high quality double-stranded DNA. We will ligate the SmartBell adapter at the site of this high quality DNA. And then after that, we will anneal and bind the DNA polymerase. And then after that, we will sequence it will actually form a circularized structure like this. And then it will actually be sequenced in repeated passes. So after that, we will actually generate, a lot of these different subreads. So all these different subreads, we will actually combine them together to form a consensus read we call the HiFi reads. So what is so special about HiFi reads is our HiFi reads are, have very high accuracy of 99.9% accuracy, and then we can generate reads that are very long, up to 25 kb. So to compare with what we have for HiFi sequencing compared to, let's say, Illumina NGS, right? You can see that for this particular study from the Precision FDA Truth Challenge, you can see that PacBio accuracy, we achieve Q33 accuracy compared to Illumina Q30. So we can say that we are as accurate as next generation sequencing right now, because I think in the past 5 years ago, people will always say, long read sequencing is not accurate. The accuracy is, less accurate compared to NGS. But, right now for HiFi sequencing, we can actually achieve up to Q33 Phred score. And then besides the accuracy part as well, you can see that, compared to short read NGS, our PacBio raw HiFi reads, are 100 times longer compared to short read NGS. So with that, right, we enable a lot of different applications and, a lot of things that NGS cannot capture on their sequencing results. Our HiFi sequencing is able to actually, deliver that. And then, of course, another thing about HiFi sequencing as well, for our WGS, right, you can actually get your methylation directly from our WGS results. So you don't need to actually, perform a separate library prep, like a whole genome, like a bisulfite sequencing pre-treatment to actually, enable you to get methylation information. For our PacBio sequencing, when we are doing WGS itself, because we are sequencing on the native DNA itself, right? And then during the sequencing run, we actually take a movie of the entire sequencing run. So compared to short read sequencing, right? So short read actually take images. In every sequencing cycle, they will take an image of the fluorescence itself to tell you whether is it a A, is it a T, is it a ATGC itself lah. But for PacBio, what we do is we actually shoot an entire movie of the entire sequencing run. So by doing that, right, we can actually detect the differences between, how fast a nucleotide actually incorporate to the complementary strand itself. So by measuring the kinetics, right, so in layman term, like, if a particular A is actually methylated, it will take a longer time to be actually incorporated. Whereas for A without methylation, you can see that it takes shorter time to actually be incorporated. So by measuring this difference in time of how fast or how slow a particular A is incorporated, we can actually tell whether that particular base is methylated or not. So by having this particular technology, we can actually, in our whole genome sequencing run, you can, besides getting ACGT, you get 5MC as well, 6MA data as well. Yeah. So coming back to, just now I talked about, how HiFi sequencing has its own advantage, right? It's highly accurate, it's long with 25 kb, and then it offers methylation data as well, 5MC, 6MA data from the whole genome sequencing data. So now we go back into, how this technology can actually contribute to, looking into cancer genomics research. So we know for cancer genomics, right, it's not only the mutations that happen doesn't only, the mutations that happen that causes cancer usually doesn't only happen on the DNA level itself, right? It's a multi-omics disease itself. So you want to look at the genomes, you want to look at the transcriptomes, and you want to look at the epigenomes as well to, give you a very comprehensive characterization of what is happening to the, the cancer cells itself. So I think the first part that I want to talk about is on the DNA level first. Okay, I just want to, on the next few slides, I will show you all some of the case studies of what, the different institutes or the different universities have been doing. They have been using our HiFi sequencing to actually advance on cancer research itself. So for this particular first study that I'm showing here, right? So we know that, for breast cancer cell line, right? A particular cell line is like, things that you grow in the lab, right? So you probably already have tested it multiple times. For this particular SKBR3 cell line, it's one of the most important models for HER2 breast cancer. So it means that, for this particular cell line, it has been used in the lab to be sequenced like multiple times on short read itself, right? So in this particular paper, they actually use our long read sequencing to actually HiFi sequencing to actually sequence through this particular cell line, and they found that there are nearly 20,000 structural variation. Most of that which were missed by short read sequencing itself. So as you know, right, short reads are 100 to the reads that they generate is from 150 to 200 base pairs, correct? So a lot of times, because of the reads are so short, right? And then they need to use like assembly tool to assemble all these reads together before they can, look at the genome itself. So because of that limitation, right, they actually miss a lot of these structural variations as well as, all this hard to sequence region, right? Repetitive regions as well. So using HiFi sequencing, because our reads are so long, right? That's why we can span through all this hard to sequence structural variations. So that's why in this particular study, you can see that with HiFi sequencing, they actually detected a lot of new variants that were not spotted by short read sequencing. And then this is also another study, that is used to actually look into infant with retinoblastoma. And then, they also have, used this, tested this particular patient multiple times using short read sequencing, but they were not able to actually identify what is actually causing this particular disease itself. So they use HiFi sequencing to actually sequence this particular patient, and they found that there were two different somatic variations or two somatic structural variations that occurs in this RB1 gene itself, and then they were able to phase the different alleles. So this is also very important. If you're using short read sequencing, you will not be able to phase the different allele, but using long read, you are able to phase the two different alleles to the paternal and maternal allele itself. And then you are also able to actually identify where the mutation is, which allele the mutation is. So they found that, in allele 1, they actually found a premature stop codon. And then in allele 2, they found a fusion gene itself. So the RB1 has fused with this particular SIAH3 gene. So with that, right, two of the different alleles are actually not working for this particular RB1 gene that's actually causing this retinoblastoma in this infant. So it helps to solve a lot of different case with our long read sequencing. Okay, and then this particular case that is more recent, you can see that for this particular case itself, they actually so if you see this IGV itself, you can see that this is the normal haplotype, right? And then the bottom is actually the tumor cell itself. So there are a few observations that you can see from this IGV itself, right? So the first one, of course, if you can actually see the big deletion at the right side of the slides itself. So that is actually causing the mutation that's happening in this particular PTEN gene. And then the second thing that you can see, right, is that there's only one allele here. So one of the allele has been lost during the mutation itself. So there's only one allele left for this particular cancer cell itself. And then the other thing, so I think the first thing that you see is loss of haplotype 1. The second thing is you can see there's a 12 kb deletion. And then the third thing you can see there's actually the promoter region for this P10 region is actually hypermethylated. So you can see that in one single sequencing run, you can actually identify a lot of different things that is not possible to actually how to say, short read sequencing will not be able to tell you all this information. Short read sequencing will probably will not be able to identify what is the cause of mutation and what is actually causing this particular cancer mutation itself. So things like the big structural deletion, short read sequencing will not be able to do that. Loss of haplotype 1, you won't be able to face on short read itself. So you will not be able to know about if there's any loss of heterozygosity or not. And then the epigenetic side as well, the methylation side as well, you need to probably run a second bisulfite sequencing to actually uh, identify this mutation. So this shows you how powerful long read or HiFi sequencing is in to actually look into all these different um, cancer markers. Yeah, and then this is also another case study on BRCA itself. So on the BRCA gene, so you can see we are comparing. So they sequence the normal cell versus the tumor cell itself. So you can see that for the tumor cell, the first thing that you see is, of course, the huge deletion, like 29 kb deletion on the first haplotype. And then the second thing that you observe is there's actually a methylated region in the second haplotype as well, hypermethylated region on this promoter region. So by looking at this, right, so actually the particular patient doesn't have any BRCA gene at all. So this is what that is actually causing, probably that particular patient to be higher risk of getting, breast or ovarian cancer itself. So the same thing, this kind of mutation, you won't be able to actually, see it on short read sequencing. Yeah, and then of course, we all know about, there are multiple long read sequencing technology in the market as well. So this is just a slide, showing some of the comparison data on comparing our competitor to PacBio on the analysis of a particular HCC 1395 cell line. So you can see that, the F1 scores are how we actually measure our accuracy and precision, right? And the higher F1 scores you have means the better performance your sequencing results is. So you can see that, with PacBio at 30 times coverage, tumor versus normal, compared to, ONT 75 times tumor versus 30 times normal, you can see that our accuracy is still much more higher. Even though, ONT is actually sequencing 2.5 times more sequencing coverage compared to PacBio, you can see that our accuracy is still much more higher compared to what ONT can actually offer. And in PacBio itself, also, we actually offer complete workflow for cancer whole genome sequencing. So we have the nanobind extraction kit to actually help you to extract high molecular weight DNA. And then if let's say you're doing, of course, whole genome sequencing, we have the SPK3 library prep to actually help you to do library prep. And then you can sequence it on the Revio or the Vega benchtop system that we have. And then, of course, on the analysis pipeline as well, we have our Somatic WDL pipeline that can actually help you to do all the variant calling, phasing, and I will show you all in the next slide. So our HiFi Somatic WDL will actually offer you somatic variant detection from cancer samples, detecting somatic and germline SNPs, indels, allele-specific copy number, somatic structural variations, differentially methylated regions, mutational signatures, homologous recombination deficiency. So you have all this different information that's actually generated. And we actually generate in an interactive analysis report format as well, which you can actually see here. So it will just go through so after our run, after you run through the HiFi Somatic WDL, they will actually generate a report like this to show you, the table, or it will show you all these filtered like SNPs, indels, and then you can actually filter through the different genes as well that you want to see. And then they will also show you the structural variations. So let's say, TP53, yeah, they will filter through for you. And then there's structural variations information as well that is generated from the table. Yeah, and I think all this different information that is actually listed down here is actually, you can actually retrieve it from the report itself. Yeah, so you don't need to generate your own report anymore. For our PacBio pipeline, we actually help you to generate this kind of report to report all the different variants, SNP, indel, structural variations, methylated regions as well in this particular report itself.
So I just want to asking the report also include annotation of the mutation, like ACMG report for somatic mutation. Like last week we have the presentation from the Qiagen. They have a database for annotate on the somatic mutation. So how about this report? Do we include the annotation database as well?
So for right now, our report doesn't include any annotation yet. So we only generate the reports out to actually help you to identify all the different, mutations that you have, but we don't annotate it. But I understand like from Kaizen, they have like, QCI and that can actually help to annotate with like cosmics as well. So it's very, I mean, yeah, so we can actually generate VCF file from our WDL pipelines, and then we can also work together with Kaizen. Let's say you need annotation, right? So they can actually import our VCF file in, and then generate those annotation from their software. But right now for PacBio, we don't offer our own annotation yet. But I think our computational biologist team is actually working on that as well. Yeah, but it's not ready yet. Yeah.
Okay. Thanks a lot.
Thank you, Dr. Loy, for your question. Okay, so the next part. So that's, I went through the genome part, like, you can actually detect all this big structural variations and repetitive regions, difficult to sequence areas on the genome using our HiFi sequencing. And then we can also, provide you phase result as well so that you can identify, where the mutation is in which different alleles. And then, of course, it comes together with the methylation information as well. So in the next part, I will go into the transcriptome. So we know, So we know the basic dogma of biology, right? From DNA will be transcribed to RNA will be translated to proteins itself, right? So any mutations that happen on the genome will actually affect what is happening on the transcriptome. But the transcriptome is even more complex. You have all these different splicing, microRNA kind of interaction as well, which will actually affect the formation of different RNA isoforms. So that's why people are also looking into, the complete transcriptome itself. So for long read sequencing itself, we can actually, fully characterize the RNA dysregulation in cancer. So looking at, RNA isoforms, fusion transcripts, and then the expression of the different genes itself, as well as the mutations that are expressed. So by using long read sequencing, you can actually look into all these different aspects for the RNA dysregulation in cancer. So I think one example that I want to show here is like, splicing events can actually, affect protein function. For example, this particular BCLXS gene, right? So the pre-mRNA will be spliced into different, you can see there's 3 different exons here. And then with the orange color exon here. So the presence of this particular orange color exon will actually form a protein called BCLXL. And then this particular BCLXL is actually anti-apoptotic, and then it will be overexpressed in cancer. Whereas for this particular one, it's for the normal mRNA. So you can see that we probably need to look into the different isoform level to be able to actually look into to differentiate between the normal cells, cancer cells, as well as looking into cancer progressions, as well as resistance to chemotherapy. So going back, right, we know that looking at isoform is very important. But why, have the existing sequencing technology not be able to actually provide enough isoform information? So we know like one single gene like this, right, will actually produce different types of mRNA isoforms. The combination of the different exons will provide you different types of mRNA isoforms, correct? So, but for short read sequencing, right, you can see that the reads that they generate are very short and very fragmented. So you can see all this, if you can see my mouse pointing, I hope you all can see it, but let me see if I can. generate the pointer. Oops, do y'all see my pointer? Okay, but yeah, I just going back to the slides just to show you, like, the short read sequencing will produce reads that are very short, right? So you need to actually, fix it up like a Lego to, tell, which mRNA isoforms is it. So short read sequencing can only assemble, 20 to 40% of the human transcriptome. It won't tell you, which isoforms you have, because you need to actually, assemble all these different fragmented reads. So in the end, you will only get a partial view of this cancer transcriptome itself. However, for long read sequencing, on the right side, you can see that we can actually sequence through the entire mRNA. So as I mentioned, right, for HiFi sequencing, we are very long. We are up to 25. We can generate reads that are up to 25 kb, right? mRNA is usually like 1.2 to like 2 kb itself. So we can actually sequence through the entire mRNA itself. So you don't need to actually, assemble them and then try to guess which mRNA isoform is itself. So we will just sequence through the entire thing. So that's why you can actually see a complete view of, the cancer transcriptome itself. So just, to show some examples of the studies for HiFi itself, going back to the breast cancer itself also, again, because, breast cancer is so well studied, right? So for this particular study itself, you can see that, for this particular, they actually, detected up to 142,000 of splice isoforms. And then 67% of these isoforms are actually, novel when they actually use our long read sequencing to actually sequence through the transcriptome itself. So there are 67% of the isoforms that was not detected on short read sequencing itself. So this show you how powerful HiFi sequencing is. And then you can see that, for the genes, for example, like ERBB2, which is, associated with HER2, right? You can see the short read only, generate, and on the gene code database itself, right? You can see there are probably only around like 20 something over isoforms. But with long read sequencing, we are able to detect up to, more than 70 new novel isoform itself. So this shows that, there are still so many things that, short read sequencing have not revealed. So, using long read sequencing, there are so many, low-hanging markers and things that you can actually detect both on the genome side as well as the transcriptome side itself. This is also another studies to show that, the alternative promoter in this particular ARD1A isoform is also a biomarker to show that the different clinical outcome in gastric cancer. So with long read sequencing, because we are sequencing through the entire mRNA itself, right? So we can actually sequence through the promoter region as well. And you can see that with this particular identification of this novel isoform, they are able to actually detect it as a biomarker for bad prognosis or worse survival. So by identifying all this biomarker as well, it can also be used for pharma to actually, try to identify some of the new drugs itself. Okay, so just now I talked about, the full-length mRNA, right? So we know that for transcriptome as well, you can also do things like single-cell RNA sequencing as well, right? So for our so in short read sequencing, let's say you're doing any 10x or parse bioscience sequencing, so you try to barcode the different transcriptomes from different cells to try to see what are the expression in all the different cells, right? However, if you're using short read sequencing, they will only be detecting the 3' end of the transcript itself. So just a few tenths of the base pairs from the 3' itself. So you will not be able to actually know about the isoform information as well. So isoform analysis is not possible. But using long read sequencing, As the same from the full-length RNA side, we can actually sequence through the entire mRNA. So we are able to actually, provide you the cancer isoform information in the single cell context. So this is also another study to show, a researcher using our single cell RNA sequencing, and then they are actually comparing between two different technology. They are using short read as well as the long read isoform sequencing itself. So you can see you can see this is a PCA plot, right, of the different cell type, and the distribution of the cell type is, you can see it's almost similar compared to short read it's similar, not almost similar compared to short read sequencing. So you just need to take the graph and you have to rotate it a bit, and then you can see that the short read and long read sequencing results are the same. And then on the right side of the slides, right, you can actually see that if you are just looking at gene-based expression, which short read sequencing can offer, you can see that the IGF-1 expression is actually similar in both the cancer cell and normal cell. But if let's say you are looking down into the isoform-based expression, you can see that there are difference in terms of expression in the class 1 and class 2 of these IGF-1 in normal cell compared to the tumor cell. So there are differences in expression. So you can see the class 2 isoform is more widely expressed in the cancer cell, whereas the class 1 is more in the normal cell. So this shows that, if you are just doing short read gene-based expression, you won't be able to tell this particular difference in the different isoform that is actually contributing to the cancer cells. Whereas if you are doing long read sequencing, you are able to look at the differences, the different isoform that is expressing in normal compared to the tumor samples itself. This is another example of if we are using accurate long read RNA sequencing, right? So I mentioned earlier, if you remember, if you are using short read sequencing, they are only sequencing like I think 50 base pairs from the 3' end, right? So if you are just if you are looking at this particular fusion gene, so this IGF2, BP2, and then it fused together with this TESPA1 fusion gene, right? So if you are using short read to actually sequence it, they will just show you, there's actually a high level of expression for the TESPA1 fusion gene. So because as I mentioned, right, so short read only sequence like the 3' end of this TESPA1. So you will only be able to detect this UTR region of this TESPA1 itself if you are using short read sequencing. But for long read, because we are sequencing through the entire gene, right? So you are able to actually tell, okay, there's actually a fusion, there's a fusion incident that actually happened. So this particular IGF2, BP2 gene actually, fused with this TESPA1 gene itself. So you can see that the bottom part, the long read sequencing can actually, assign all the transcript to this particular novel fusion. And then, of course, on our long read RNA sequencing itself, also we can actually detect somatic mutation as well. So because I mentioned about, our accuracy is up to 99.9% accurate, right? So you can actually accurately call all this, somatic mutation on the RNA level as well. It's not only on DNA, but you can also do variant calling on RNA. And then there's another group that actually looks into the neoantigen receptor itself. So the neoantigen field has always, just looking at SNPs itself, right, because of short read limitation. But large structural variations and like isoforms are also likely to be more immunogenic as well. So by doing this particular study, they can actually, find new using long read RNA sequencing, you can actually discover more robust neoantigen, to actually develop cancer vaccine. Yeah, so in terms of the RNA side as well, we do have a complete workflow. So like from the sample prep, and then we have this Kinnex library prep workflow that I will show you later. What is Kinnex library prep? And then after you prepare the library on Kinnex, you can actually sequence it on the Revio or the Vega instrument. And then once you have the isoform data, we have the pipelines available on SmartLink itself that can actually help you to do isoform discovery, tell you, how many novel isoform is there, how many known isoform is there. And then if you want to look at fusions, we have PB Fusions tool as well that can actually help you to identify fusion. So I mentioned about our Kinnex library prep workflow, right? So as I mentioned, for RNA itself, RNA is quite short, right? It's only like 1.2 or to 2 kb long, usually, correct? So as I mentioned, if you all remember how long was HiFi sequencing? So HiFi sequencing is, we can actually generate reads that are up to 25 kb long, correct? So if we are just using each well to actually sequence like 1 or 2 kb of the mRNA itself, we will be wasting a lot of the capacity of the HiFi sequencing. So we are only sequencing 1 to 2 kb, right? So there's, like 23 kb capacity that is wasted itself. So what Kinnex do is we actually combine these multiple cDNA together. What we concatenate all these multiple cDNA together into one single long read, and then we actually sequence it on our sequencer itself. So we combine, up to like 8 mRNA together on a full-length RNA kit. And then on the single cell kit, we actually combine up to 16 different transcripts so that, we can actually increase the throughput that you actually get from a sequencing cell itself. I mean in the sequencing smart cell itself. And then by doing that as well, we increase the throughput, the price will also go down. And then this enable us to actually do, gene expression studies as well. Yeah, so this is, another tool that I mentioned earlier on, PB Fusion tool. They can actually, accurately call where the breakpoints are. So you can accurately identify where the exact breakpoints are using this particular tool. And then they can also form this circular diagram to actually show you the different, where are all the fusions combining between the different genes.
Just to ask one question before you move to the next section.
Yeah.
For the sequencing RNA-seq, can we detect on the RNA modification like the SIG-MA, some modification like DNA methylation, but in the RNA?
Yeah, so I think we can. So there are customer developed protocol that they did some pre-treatment before sequencing it through our PacBio sequencing. So we do have some publication where some customers actually develop some pre-treatment protocol. So they change some of this like M6A modification to an alternative base, and then after that they sequence it on PacBio itself. So by doing that, we can actually detect DNA modification. Yeah. So compared to our competitor, I think Dr. Lloyd, you want to. What our competitor's strength is, of course, they say they sequence through like native RNA, right? So that actually enable them to actually look at the modification part. But I think it still depends on the application of what you want to look at. If let's say the modification that I think they are generating is also just a probability right now, like they just tell you maybe 60% this particular base has a modification, but it's not like 100% sure yet. And then I think our strength is also in terms of, let's say you want to look at RNA variant calling. So our bases are, of course, much more accurate compared to what they can actually offer.
Yeah, thanks a lot. And how about the Kinnex kits with the RNA-seq from the HiFi PacBio sequencing? Can we it will adapt for all the other single cell platform like the 10x Genomics or the other platform as well, or only specific for some of the platform for the single cell technology?
Yeah, so our Kinnex single cell platform, the Kinnex single cell kit is optimized on the 10x 10x platform, and then there's also a new protocol that came out for Parse as well. So if you're using Parse, it's also compatible with our Kinect single cell workflow.
Okay, it means that we don't need to convert anything, just directly use with the part.
Correct. There's a protocol that we actually have that you can actually use directly.
Okay. Okay, thanks a lot.
Okay, so I'll go to the next section.
Hello.
Hi.
Yes. Thank you, Raylan, for your express the brief video express now. I want to ask question for the slide in ICA 1A. I don't remember at what do you want to share?
Which one? Sorry. Do you remember which slide?
Doctor Lợi Lưu send slide to me. Cái phương pháp mà ICA1A là cái phương pháp đó như thế nào, có thể chia sẻ lại.
Cái slide số mấy á, anh nhớ không ạ?
Anh không thể nhớ, nhưng mà cái
anh gửi em cái tên đi để em dịch lại.
Okay.
I'm sorry, like
A moment, I'm trying to translate his
Okay,
question
okay.
to you. Okay. Anh gõ tên cái công nghệ đó vô đi rồi em hỏi anh Ray cho. PD1A.
Xin lỗi anh, em cũng không biết công nghệ đó là cái gì với anh. Xin lỗi. Dạ.
Trong slide luôn hay sao anh? Hồi nãy em cũng không thấy nó.
One slide in the middle online. On slide.
All right.
Next
Ray, can you re-explain about like?
Yeah.
Can you please re-explain about the technology DTD1A? I don't know which slide, but he want you to re-explain about the technology name DTD1A. He typed in the Zoom chat box. Can you see it?
The TD1A.
Yes, I'm not sure. Where is it?
Do I remember? Is there
Ở trong slide luôn đúng không anh ơi? Hồi nãy
Yes,
hình
yes.
như
In on slide.
about mà về cái
Before TF DNA, ctDNA. Before slide.
Before slide?
Yes.
please return until
I'm trying to find where the 1A is.
anh đang move lại đó, cái đến slide đó thì anh bảo dần nha anh, em cũng không nhớ nằm đâu.
Here.
Okay.
Here.
Next slide.
Next slide.
Okay, this one. Sorry.
ID1A.
Yeah, your question is
not really understand about like the whole slide here. So can you please to re-explain again for him?
Oh, okay. So I think the main message from this particular slide is so they were actually looking at gastric cancer, right? So they actually use our long read isoform sequencing to actually look into this particular gastric cancer. And then they found two new isoforms for this particular gene. This AR1D1A gene. So they found that there was two new particular isoform, and then this particular isoform, right? One of them is actually have a truncated. Actually uses a new novel promoter. So this particular novel promoter actually resulted in a truncated protein. So this particular novel isoform, right, actually truncate the protein through the usage of a particular alternative promoter. So if you are using short read sequencing, right, you won't be able to actually detect this particular novel isoform itself. But by long read sequencing, because we sequence through the entire mRNA, so that's why you are able to actually detect these two different isoforms for this particular gene. And then they were actually looking at this particular, I think this is what we call the Kaplan-Meier curve, right? That actually shows, I think, the survival of a particular patient itself. And then you can see that for this particular novel promoter, it actually the survival rate for the particular patient actually decreased by a lot. So the number of days actually are shorter compared to this particular if the particular patient has the isoform with a known promoter, they can actually survive longer. But if let's say they have an isoform with this novel promoter, it's a biomarker for actually a worse survival. So means the patient particularly will die earlier because they have this particular isoform with this novel promoter. So which means that by being able to look at the different types of isoforms, we are able to actually identify this kind of biomarkers that will actually help to predict predict the prognosis, or tell the prognosis for a particular patient itself. So by having this technology itself, pharma companies, they can also look into this kind of biomarker, right, to see whether can they develop a particular drugs to actually target this particular isoform, so that it can actually help to prolong the life of patient itself. So yeah, so I think this particular study is one, just want to say, because of them using a long read sequencing itself, they are able to see new isoforms that can actually predict prognosis of a particular patient.
Dạ anh ơi, anh ok chưa?
Okay, I
Dạ,
agree that.
tức là tại vì em giải thích ngắn ngắn lại xíu ha, tại vì anh nói tức là cái long read này nó giúp mà phát hiện thêm những cái isoform mới, tại vì nó giải từ đầu 3 phẩy đến 5 phẩy thay vì short read thì nó không có hết, nó chỉ nhìn được một phần của những cái transcriptome này thôi, còn cái này nó phát hiện hết, cho nên là người ta mới phát triển thành biomarker mới để phát triển thuốc mới, thì cái đại ý là vậy ạ.
Vâng, cho anh nói tiếng Việt em dịch giúp đó. Tức là
Dạ,
tại vì
dạ.
cái này nó là một cái marker lạ nên anh quá chú ý, nhưng mà nói chung là cũng sẽ có được nhiều cái mở rộng cái hiểu biết hơn, nhưng mà thực sự là nếu ở Việt Nam thì hình như chưa có làm được cái marker này đâu.
Cái đó chắc là anh Ray sợ anh không giải thích được anh, cái này anh chỉ nói về mặt công nghệ thôi, còn những cái ứng dụng của cái này như thế nào thì sẽ là những cái nhà nghiên cứu hoặc là những bác sĩ anh, còn như cái ý mà anh muốn nói ở đây tức là
Không,
nó
ý anh muốn nói là hiện tại ở Việt Nam chưa có cái công ty nào nó làm cái marker này á, nên anh chú ý vào là anh nhìn thấy cái giới thiệu cái marker hơi lạ nên anh muốn cho nhìn rõ hơn. Rồi anh sẽ ghi nhớ là lần sau anh sẽ có cần thì anh sẽ đòi hỏi. Rồi cảm ơn.
Dạ cảm ơn. He just asking, do you know any Vietnamese company that already developed the biomarker that you mentioned in the slides?
I'm
do you
not
know?
very sure.
Anh không biết là anh không không chắc rồi Đắc ơi, chắc là anh chỉ nói về mặt công nghệ thôi, còn đương nhiên là chắc chắn sẽ có, nhưng mà ở Việt Nam có không thì anh không biết. Dạ em cảm ơn ạ. Thank you Mr. Ray.
No
You can
problem. But I think
okay.
I think like the question is valid lah. I mean we know this kind of technology is new, right? And then there are many cancer type that are, maybe unique to the Vietnamese population itself. So
Ừ.
so like what I mentioned, right? Like in the earlier on, I shown a few of the study itself that, for things like even breast cancer, that was very well studied, right? But it was well studied on short read itself. But if we can apply long read sequencing to actually do both genomic DNA sequencing as well as transcriptomic DNA sequencing, there will be many, new discovery or many new things that you can actually, I mean, to be able, just looking back at any of the cancer samples that you have, if you do long read sequencing on it, you will be able to detect a lot of the new things that you might not be able to actually see in short read sequencing itself. Yeah.
Thanks a lot for your information. Dạ,
Ừ.
dạ ý anh nói đây là cái này giống như là long read thì sẽ giúp có nhiều thông tin hơn, có thể là sử dụng những cái mẫu cũ trước đây mình bằng short read, mình giải short read mình không có biết được những cái thông tin quý giá đó, bây giờ mình dùng long read thì mình khám phá thêm nhiều thông tin hơn đó anh. Thì cũng giống như nãy em nói từ đầu á, còn cái việc mà ứng dụng những thông tin đó, tìm hiểu được đó như thế nào thì là chắc là thuộc về chuyên môn của bác sĩ hoặc là những cái nhà nghiên cứu về ung thư á. Dạ em cảm ơn ạ. Thank you. You can move to the other slides.
Okay. Let me go to the next section. Okay, so I should continue on. So I covered both genome and transcriptome, right? And I think during the genome part as well, I talk a little bit about how we actually our whole genome sequencing data is able to give you methylation information as well, where you can see if a particular gene is hypermethylated or hypomethylated, which will eventually affect the expression of the gene, right? So the next part I will just talk about, just show more example for uh, epigenomes itself. So I think this is just to show you an example of, again, a particular tumor specific hypermethylation of this HOXD9 gene, so that's associated with EMT, right? So you can see that this particular tumor samples is hypermethylated not only in the promoter region, but it's also in the gene itself. You can also see regions that are hypermethylated itself. So these are additional information that you can actually get from just whole genome sequencing of a particular tumor sample. And then another new advancement from PacBio last year was a pre-treatment process we call the FiberSeq. So the FiberSeq enable the additional information we call the chromatin accessibility. In addition to genome, metallome, and transcriptome, right? With this particular FiberSeq technique, so what it does is, like, we actually have a metal 6 transferase enzymes that it will actually convert some of this uh, DNA that is actually accessible. There is not, that is not in the chromatin. So if you see all this big round thing, these are all, like, chromatins, right? Which are, they will entangle the DNA onto it, and then those region might not be accessible, and then it will not be able to express, correct? And then by doing this particular process, we are able to actually identify what areas of the genome is actually accessible, and by doing that, we can actually detect all these M6A markers itself. So you can see that it's not only giving you 5MC information, ATGC. So with FiberSeq, it will actually give you this M6A base information also. So you can see this particular study that was done by University of Washington. So they developed this particular FiberSeq technology, right? So they run a particular sample, through FiberSeq, and then through our normal library prep process, and then they also prepare a second library which is on RNA, and then going through the Kinex library prep that I mentioned, right? And then what they do is they pull the two different libraries together and then sequence on one Revio smart cell. So just one smart cell, yeah. So just on one Revio smart cell, they are able to actually look at the genome information, the metallome, the chromatin epigenome, as well as transcriptome information itself. So in one single Revio smart cell, you can actually get a multi-omic data already. So this is an example of the IGV that you will actually get after you do this particular study, right? So you will be able, in one single Revio smart cell, you're able to look at the genetic variants. So you can see on the top here, there's genetic variants, and then you can see the CPG islands, the CPG metallome, and then the chromatin epigenome to see the chromatin accessibility, and then, of course, the transcriptome as well. And then of course, the most amazing thing is like you can see two different haplotypes. So the paternal and maternal allele. So the results are phased. So you are able to see, the four different ohms, and then from there, they are able to actually look into they can actually associate, what is happening on the phenotype event compared to what is happening on the molecular level. So they are able to correlate, correlate between, for example, the particular patient has a bilateral retinoblastomas, right? So they are able to actually know to say that, it's because of the X chromosome inactivation that's happening on the RB1 locus that is enabled by the chromatin epigenome. So this is the reason why the particular patient has retinoblastomas, So in one single smart cell, you are able to actually generate all this different information to be able to, better characterize what is happening on the phenotype level. So this just shows you how powerful HiFi sequencing is right now. Any question? I think that is on the epigenome part. So I think for the epigenome, besides 5MC, we can generate 6MA as well with the FiberSeq data. And then, with one single smart cell right now, you can do genomes, transcriptomes, as well as epigenomes to actually, provide you a complete characterization of that particular patient, whether is it rare disease or cancer genomics itself. Okay, so I think, should I continue, or if there's anyone that would like to ask any questions?
I received one question in Vietnamese, but it's too long, and I think let me translate for you, but I'm not sure I translate correctly. Can you see in the chat box? I just summarize his questions. He has 4 questions here. So can you see on the chat box, Mr. Ray?
Yes, I see.
Yeah, one by one, please. Thank you.
Yeah, so I think the first question is like whether the application of PacBio isoseq is economically feasible in Vietnam, right? I think for right now, if you do keep track of PacBio sequencing itself, right? So from PacBio sequencing itself, our price of sequencing has been decreasing every year. It's actually decreasing because of the new technology. For example, when we launched the Revio, we can actually run, a 90 GB on one smart cell itself. So we are able to generate genomes at, 30x genome at 1,200 USD at that time. And then after that, we also launched a new chemistry like Spark chemistry itself. So the Spark chemistry also, increases the throughput of the Revio itself, and then, further bringing the sequencing cost down. So I think for PacBio isoseq right now, and together for isoseq itself, we have the new kit on Kinex itself. So with the Kinex itself, the Kinex kit will actually help to increase the throughput of the isoform I mean, the throughput of the RNA reads that you actually get from one single smart cell. So because of all this different technology, right, the isoseq is getting more and more economically flexible. That's why you see, I think in this 2 recent years, there's so much more isoform related publication that is being published nowadays.
Thank you. Let me translate first. Anh ơi, cái câu đầu tiên là hỏi nó cái isoseq nó có hiệu quả kinh tế hay không á, thì anh nói là nếu mình theo dõi PacBio từ lâu á thì mình cũng có thể biết được là giá thành ngày càng giảm dựa trên cái team R&D của họ, họ làm rất là mạnh luôn. Thì ví dụ như là trước đây giải trên cái Rebel System á, thì bây giờ nó có những cái hệ thống khác thì nó sẽ làm giảm giá thành nè, thứ nhất. Thứ hai là họ cũng phát triển thêm một cái chemistry, em nhớ là tên gì, nó cũng làm tăng cái throughput của cái tức là cái thông lượng mình giải của cái hệ giải trình tự. Cái thứ ba là nãy anh có nói cái Kinect kit á, tức là ví dụ như là cái giải HiFi của anh là từ 25 đến 30 KB đi, còn của mình nó ngắn hơn thì mình có thể dùng cái Kinect đó mình concat lại, tức là mình ráp nó lại để mà mình giải được nhiều mẫu hơn, thì cái giá thành nó sẽ giảm xuống rất là nhiều. Thì ý anh muốn nói đây là cái team R&D đang làm, nói chung phát triển rất là nhiều thứ để làm giảm giá thành xuống từ nãy em nhớ không lầm là giống như từ $1,000 mà xuống còn $100 vậy đó, thì nó sẽ rẻ hơn rất nhiều, nhưng với điều kiện là mình phải gom đủ mẫu lại, và đương nhiên là mình cũng phải có cái chiến lược vun mẫu sao cho nó hợp lý á. Thì cái đó em trả lời thêm, còn ý anh nói đây là giá thành đang giảm, nhưng mà nó chỉ rẻ khi với điều kiện mình giải được nhiều mẫu cùng một lúc. Ví dụ mình có cái chiến lược gom đủ mẫu trong cùng một thời điểm hay gì đó, mình giải thì nó sẽ rẻ hơn rất là nhiều ạ. Dạ em cảm ơn ạ. Okay, please, the next questions, please.
The next question is like, I think he's asking about whether, have we done isoseq on fresh or FFPE tissue samples itself, right? So I think the crucial questions about RNA is very sensitive, right? So we always, I mean, on the FFPE samples, I think even on the short read side as well, we know that RNA is very sensitive. If you go through like FFPE process and everything, the RNA will be degraded itself. So I think there's not a lot of studies for isoseq on FFPE. There are more studies for isoseq on fresh frozen tissue or like blood samples. But I think for FFP tissue samples, right, I will talk about a new technology which is on my screen right now, which is called amplify. So on the DNA level, we are actually enabling some of this long read sequencing for FFP tissue samples, which is very exciting.
Thank you, let me translate. Anh nói là cho cái mẫu FFPE hay là mẫu mô tươi mà anh nói á, thì tại vì mọi người cũng biết là cái RNA nó rất là nhạy cảm, cho nên là cũng không có quá nhiều nghiên cứu mà về cái FFPE tissue này, mà nhiều hơn là cho mẫu máu hay là cái mẫu khác. Nhưng mà cái FFPE này thì anh sẽ đề cập đến cái công nghệ mới mà anh sắp nói nha, thì nó sẽ giải cái mẫu FFPE nhiều hơn ạ. Thank you. The next one, please.
Okay. So I think the third one is about whether are we developing therapies based on specific RNA isoforms, right? I think there are like a lot of different applications. I think a lot of the different publications that, why they want to, look into new isoforms or like new biomarkers. A lot of times, their final goal is actually to develop, a ASO or like, a vaccine kind of thing for, downstream work, whether is it on the rare disease side or the cancer side itself. So there are ongoing efforts. But whether, as we know, all these therapies need to go through clinical trial and things like that. So it will probably take a few more years before they are able to generate out.
Thank you. Dạ cái câu này thì cũng anh cũng nói là đương nhiên là có rồi ạ. Okay, thank you. The last one, please.
Yeah, so I think the fourth one has Vietnam plan to build a cancer isoform atlas. I would be happy to support if there's a research team that is looking into building a cancer isoform atlas in Vietnam itself. So we know, what I know is like the Japan team. I mean on the Japan side, they do have this cancer genome atlas program that they are also using our long read sequencing to actually support that program. I think in the Singapore side, they have this like they have human cell atlas that is trying to profile every single cell's expression. I mean, in every cells like in healthy patients, I think. But they are also exploring long read sequencing right now to actually go into isoform level information.
Dạ, thank you. Dạ cái này anh nói là hiện tại thì ở Nhật Bản với lại ở Nhật Bản thì có cái Cancer Genome Atlas. Dự án này thì là bên ảnh cũng đang hỗ trợ ạ. Còn Việt Nam thì nếu mà có thì ảnh cũng rất là vui để mà, đương nhiên là PacBio rất là vui để mà hỗ trợ. Cái này nó cũng giống như win-win đó anh, tại vì hàng năm á, nói chung là những cái sự kiện của PacBio mà em đi mà em biết á, thì giống như là họ đều mời những cái bên họ giới thiệu công nghệ, nhưng họ mời những cái cơ sở nghiên cứu lớn trên thế giới đó là trình bày những cái mà họ đang nghiên cứu mà ứng dụng cái công nghệ của PacBio, thì nó là win-win thôi, đương nhiên là họ rất là muốn hỗ trợ rồi ạ. Dạ không biết anh còn câu hỏi, anh hài lòng với câu trả lời không ạ? Anh Linh ạ. Còn nếu mà khó quá thì lát nữa thì lại về mình hỏi thêm cũng được ạ.
Rồi, xin cảm ơn ạ. Yeah,
Dạ.
thank you.
Yeah, he satisfied with your answer. Thank you. Can you please continue?
Okay. All right. I will continue. Let me close the chat box first. Okay. So the next section is to talk about some of the advancement from PacBio as well. So we just recently launched a new protocol, which we call it amplify. So what does amplify means is like we are amplifying the genomic DNA for library prep and HiFi sequencing itself. So if you remember the previous part where I talk about our whole genome sequencing workflow, right? We are actually, sequencing through the native DNA. So there's no PCR process or there's no amplification that happens during our normal library prep workflow. But for now, because we know that there are certain, challenging sample type, or sometimes people just don't get enough sample DNA to go through long read sequencing, right? So that's why we actually introduced this particular protocol called amplify. So for amplify itself, we can actually perform sequencing on Revio and Vega with as little as 1 nanogram of gDNA. So I think like what I mentioned, right? A lot of times, why people don't do long read sequencing is because they don't have enough samples. Because I think for even our Spark chemistry right now, we will require you to have at least 500 nanograms of genomic DNA before you can actually proceed for proceed for library prep and sequencing. So this has been 500 nanograms is already like an advancement. If 5 years ago you asked about this question, I think 5 years ago you probably need micrograms of gDNA, like maybe up to like 5 micrograms for one single run. But right now, because of the new chemistry as well, we managed to reduce our requirement down to 500 nanograms of gDNA for our normal workflow. And right now, with this particular amplify protocol, we can actually support as low as 1 nanogram. So you can see in this particular slide, for PacBio protocol for amplify, you need around 1 to 50 nanogram of gDNA. So there's a amplification step at the start where we actually use KOD extreme hot start DNA polymerase, a high fidelity DNA polymerase, to actually do amplification at the start. And then we will also use the twist UDI system as well to actually, prevent any PCR duplicates, to identify any PCR duplicates. So, and then after that, you have these two processes at the start, and then the downstream workflow is the same with our normal library prep workflow. And then once you have your library, you can sequence it on the Revio or the Vega system. And then we also have, our Smart Link will be able to actually help you to demultiplex and to identify the PCR duplicates and also remove them from our HiFi BAM. And then after that, you can use back our existing third-party tool, like what I shared earlier on, like the Somatic WDL or like the normal WDL, to do all the variant calling, facing, all those different tools. You can actually use the same tools for this workflow as well. So with that, right, with this particular protocol which can actually support less than 50 nanogram of gDNA. So now we can actually support things like sequencing small body organism, for example, like small insects. Maybe you just want to sequence one particular insect, you can actually use this protocol, or things like metagenomics as well. Or of course, I think one of the most interesting topics for cancer genomics is FFPE tissues. So with this amplify itself, we can also support FFPE tissues, which I will show in the next slide. So we know FFPE samples are very challenging, right? It's going through like the formalin treatment itself. It's highly degraded, and people always think FFPE DNAs will usually be very short. So we tried to sequence a few different samples with our amplify protocol, and you can see that if you see this distribution chart, right, this is the read length of the DNA that you get from an FFPE sample. So you can see average is around average. You can see the average read length for FFPE tissue is probably like 2 kb, and then with also like long fragments up to 8 kb itself. So it's generally not as complete as 15 to 25 kb like what we do for fresh frozen tissue or blood samples. But there are still fragments that are very long as well. I mean long in terms of like 2 to 8 kb as well in the FFPE samples itself. So with that, you can see that these are just some of the metrics that we generated from our internal run. So you can see that the HiFi yield that you usually get from FFPE samples will be lower because the read length is lower itself. But what I want you to focus is on the read quality itself. You can see the read quality that we get from FFPE samples are still up to like Q50 Q score itself. And then so what does it mean that like what's the advantage having to do long read sequencing for FFPE samples, right? Of course, the first thing, like what I mentioned earlier on in my slides, is you can face the genome. You are able to face the paternal and maternal allele, and then you are able to actually identify where the mutations are. Which is the mutation in the trans or in the cis allele itself, and then whether a certain mutation is it in the same allele or is it in two different alleles. So that will also impact the expression of a certain gene, right? Correct. And then of course on long read itself, we are able to, like what I mentioned, we are able to sequence through all this like giant brick deletion, structural variation. So with that, our amplify with HiFi sequencing can enable you to actually do a lot of this different give you a lot of these findings through this amplify, which enable you to actually run on FFPE samples. Yeah, so that's the latest protocol that we actually enable on the cancer genomic side. Is that now it's now on HiFi sequencing, we don't only can support like fresh frozen tissue, hematological cancers with blood samples. So with Amplify itself, we can also support FFP samples also for cancer genomics. Okay, maybe I pause a while to see if there's any question.
Dạ, mọi người có câu hỏi nào mà? À, we have no questions. Okay.
Okay, if no questions, then I will continue. All right, so the next section so I talk about how HiFi sequencing is helping you to do all this comprehensive characterization of a cancer sample, right? Looking at not only the genomes, transcriptomes, epigenomes. You get so much more information from one single smart cell. You can do all this multi-omic study on HiFi sequencing to actually completely characterize cancer genome itself. So looking at so many more data compared to if you're doing like short read sequencing, you probably just get DNA and you can only look at SNPs and indels. But on HiFi sequencing, you can look into like all this, besides SNP indels, you can be looking into structural variations, repetitive regions, and then pseudogenes, and then you get it in phase. You can actually phase those different the different alleles, and then you also get methylation information as well from HiFi sequencing. Okay. So the next section is to talk about, our SBB sequencing side, right? So we know there are still certain applications, for example, like liquid biopsy, that may not be, very optimum for HiFi sequencing. So that's why, in PacBio itself, we have both short and long read as well. So for application that, requires higher sensitivity, we do have SBB sequencing that can actually help to actually support those type of application. So HiFi sequencing is more like, to provide you a comprehensive characterization of the particular cancer genome. But for application that requires high sensitivity, we have SBB sequencing to actually help to support those type of application. So for SBB sequencing by binding technology itself, right, we actually have our short read sequencer, which is shown here, called the ONSO system. So the ONSO system, what's the value of our ONSO system is like we can generate bases that are, more than 90% above Q40. So I think the general benchmark right now, I think for most of the short read sequencing is probably like Q20 to Q30. But for our ONSO system itself, we promise our customers that we can actually generate more than 90% bases above Q40 plus. Yeah, so I think so what's the advantage, or like I mentioned about, we can generate reads that are highly accurate, Q40 plus. So what does it mean, right, to the application itself? So with greater accuracy, it actually enables you to have greater sensitivity itself. So because of the high accuracy, right, you can actually have higher sensitivity to detect a particular, for example, a mutation in ctDNA. And we know that, usually for the mutations that happen for ctDNA, the allele frequency is very low, correct? Probably like 1% or even lower than 1% itself. In ctDNA, it might be like 0.1%. So if your sequencer is not sensitive enough, you might miss some of this mutation itself. So that's why, with greater accuracy, we will be able to achieve greater sensitivity. And then you can see that, in terms of cost as well, because of the greater accuracy, which enable you to have greater sensitivity, right? So in order to achieve similar performance like the other short read sequencing, you can actually reduce the coverage required. So you don't need to sequence so much to actually achieve similar sensitivity. So by doing that, you're able to reduce your cost as well, which I will show you some of the case study later on as well. Yeah. So with higher sensitivity, right, like this means that you can actually, detect certain cancer type earlier, or things like, monitoring of cancer, like, minimal residual diseases. We can actually improve the performance of the test itself. And then, of course, with SBB technology, we are able to sequence through like difficult to sequence region, like microsatellites as well. And with that, we can provide you greater confidence in your results. So yeah, just to show you all some of what is the basis of sequencing by binding, right? So we know the conventional method that most of the labs are using are still based on sequencing by synthesis, correct? So what sequencing by synthesis works is like you will have a original strand, and then you will have a new strand that is actually we are building on the new strand, and then each of the sequencing cycle, we will actually incorporate a new base with a fluorescent signal. So maybe G is blue, A is red, T is green, and then it will incorporate to the complementary strand, and then it will emit the particular signal. So after that, after the camera record the signal, then it will actually cleave the fluorescent away as well as the blocker away, and then this particular cycle happens again. But what are the disadvantage of this is like what if the polymerase doesn't cleave off the fluorescent, And then there's also this particular, additional, uh, section here, which might also cause like molecular scarring as well. So all this, will actually increase the noise that is happening during the sequencing cycle. So by having higher noise, you will actually decrease your accuracy. So but for sequencing by binding, for SBB, you can see that, what we do is we separate the interrogation step and the incorporation step. So for the interrogation step, you can see that we use a nucleotide with a blocker, and then we will actually, the base with a fluorescence will actually bind temporarily with the original strand, and then after that we will take the signal, and then after that we will wash away the nucleotide with the fluorescent, and then after that we will activate the base itself by removing the blocker, and then after that we will attach nucleotide a new nucleotide without the fluorescent and with a blocker itself. So by doing that, we will not have the problem of, whether, the leftover fluorescence is not being cleaved away, or like there's a molecular scar nucleotide. So we don't have those problem itself. So that's how we actually, increase our accuracy. Yeah, and just to show you some of examples that are being done internally as well. So you can see that for most of our runs, 100% of our bases are above Q40, and then this is an example run that actually we can actually achieve up to like 85% of our bases to be Q50. Yeah, so just to reiterate again, some of the benefits is like we can actually sequence through the difficult repetitive regions, and then there's no index hopping as well because of our different technique compared to SBS, and then there's also low duplication rate, and then you can also lower your input requirements, and finally, of course, like what I mentioned, because of our higher sensitivity, you can sequence lesser, so you can actually reduce the costs. This is just an example to show you, compared to SBB and SBS in a region with homopolymers Lang. So you can see that, with our SBB technology, we can support, up to like, 25 similar nucleotides, and it's still going strong compared to, SBS where the performance is already degrading with the increased Lang in the homopolymer. So this will help in terms of like, application like MSI, mutation rate analysis, or like things like poly A tail sizing. Yeah, so this is, I mentioned about, why SBB has negligible index hopping and lower duplication rate, right? It's because that we use this rolling circle amplification protocol for template preparation or cluster generation. So that's why we will not have index hopping issue or or duplication rate. Yeah, so just to mention again, to further elaborate again, because of our lesser duplication rate itself, the number of redundant reads that you get for SBB is also lower compared to the other technologies. So there's no wasted sequencing that happens. And of course, like what I mentioned, with SBB, right, you actually need to sequence less. So for example, for this, I will show you example case studies later, but just to show you some of the like liquid biopsy assays, right? For example, if you run on a SBS platform, their recommendation is probably asking you to run up to 25,000x, right? However, because of our also higher accuracy, right, you can actually lower down the coverage, maybe down to fourfold itself. So by lowering down the sequencing coverage, you are able to actually multiplex more samples on the sequencer. So by doing that, we can actually reduce on the per sample costs. So just to I talk about how we can actually sequence less, right? But you probably want to see like real data, correct? So we have this particular head-to-head comparison using Serasic ctDNA reference. So this particular Serasic has known variants, so they have 19 small variants, and then we run it through two different technology, the SBS and SBB. Yeah, so so if you see the image on the left, right? So if you see on this particular SBS results, and we actually spiked like 0.1% VAF of the mutation together with the wild type. You can see the results that you actually get from the SBS chemistry. Excuse me, sorry.
You can go to have a sip of water. We can wait.
Probably. Okay.
Okay, thank you.
Yeah, so if you see SBS, you can see that the results that you get from the SBS, right? Even though the mutation that we spike, if you see the results here, you can see that there's 7 count of A, 10 count of C, 10 count of G, and then T is the wild type, right? If you are just looking at this particular result, you wouldn't know, what uh, the spike mutation is, or like, what is the real mutation. However, if you look at SBB data, right, you can see very clearly, there's 0A, 0G, and there's 13 count of C. So, by doing that, you can actually accurately tell okay, the mutation is actually C. So this shows you, comparing to SBS, which is a bit noisy, for SBB, you can actually confidently say, what are the true variant is. And then this is also another comparison, so if let's say, we are sequencing, comparing between SBB and SBS at 12,000x coverage, 12,000x coverage, you can see that in like 0.05% VAF, you can see that the SBB sensitivity is much more accurate compared to what SBS can offer. And then at 0.1%, you can see that the result is so precise, it's only one line. And then we also tried to actually downsample the SBB to see that, what is the lowest coverage that we can go that is, equivalent to the performance for SBS. So you can see that, for example, for 0.05% VAF, at 6,000x, we are on par with 24,000x of SBS sequencing. And then the same thing on the VAF of 0.1% as well. So we actually need 4-fold less sequencing compared to the SBS, and then without the UMI as well. So that's why, I mentioned about, you actually have to sequence because of a high accuracy. You don't need to sequence as much as you sequence on SBS to actually get the same sensitivity. So by doing that, you can actually multiplex more sample in one single flow cell, and then that will actually help you to reduce the costs. Yeah, and also talking about the noise of the sequencer itself, right? So if you do look at this particular comparison between SBB and SBS, right? So you can see that for wild type for SBB, our the wild type is, you can see that the precision, the sequencer is very precise, that, you don't have any noise in the wild type side. But however, if you look at SBS, right, you can see that even the wild type, you still are getting some of the signal itself to show you some observed allele frequency. So if you are getting results like the what you are getting on SBS, right? So the threshold that you have to put uh, is going to be very high. You cannot be able to tell, whether a particular sample is actually a wild type or sample. If you do look at, anything that is below 0.1%, right? You wouldn't know whether is it real with the mutation or is it really the wild type. But for SBB, you can see that it's very clear. The wild type is totally separated from the samples with spike mutation itself. So that's why you can actually, confidently tell whether whether for this with SBB, it's much more accurate. You can confidently tell which one actually has the mutation compared to the wild type. And yeah, and also, on the SBB itself, you can see that the results that you get is closer to what we actually spike in as well. So for example, this study, we spiked in 0.1% I mean mutation, right? So you can see that at SBB and SBS, our observed VAF is much more closer to the true positive or the 0.1% VAF compared to SBS. Yeah, so finally, just to show you all some of the, case studies that what other people are using our ONSO for. So as I mentioned, right, ONSO is very, it's the advantage is of course the accuracy of the sequencer itself, which enable a lot of highly sensitive application. So for example, liquid biopsy. So we work together with TGen. So you can see TGen is actually, using our ONSO system for liquid biopsy for multi-cancer early detection. And then, the pilot study, so you can see that on the left side, right? So that's the pilot study that they have done. And they saw that, the Q score for ONSO is, above Q50. Like most of the reads are, above Q50s, Q50 compared to, the SBS sequencer. And then we also work with McGill as well, that is using it for early detection of ovarian and endometrial cancer. And they are using a targeted panel as well. And you can see that the IGV view that the PacBio results is much more cleaner compared to what Illumina, you can see on Illumina, right? So the Illumina side, you can see there's so many, noise in the IGV itself. Yeah, and then they also saw that there's the error rate is also much more lower in the SBB sequencer compared to the SBS. Yeah, so I think just to summarize the value of SBB, right? I think from this particular review paper, they actually showed that a key challenge in the early detection of cancer is usually caused by a particular technology that is not sensitive enough and specific enough. So if let's say we have a technology that is like ONSO that has high sensitivity, and with high sensitivity it give you high specificity as well to confidently call how much percentage of the variant is there in a particular sample. This can actually help to disrupt the early cancer detection technology segment itself. So with that, I think that's the last of my slides. So, just to summarize again, for SBB sequencing, we have an ONSO system that can actually generate reads that are 90% above Q40. And with that, with high accuracy, you can actually achieve higher sensitivity, and you can sequence less to actually save on cost to actually achieve the similar sensitivity like the SBS sequencer itself. Yeah. So thank you so much. If you all have any question on the short read sequencing side, please let me know.
Thanks so much for your sharing. Your presentation is very informative. Mọi người ơi, mọi người có câu hỏi gì không ạ?
Chị có thể đặt câu hỏi được không em ơi?
Dạ chị, cô Diệp hỏi trực tiếp tiếng Anh lên chị.
Hỏi trực tiếp tiếng Anh của mình nó hơi dở. So hi, nice to see you again.
Hi, hello.
Yeah. Actually, very nice presentation. Thank you, and a lot of information. So I can see that every problem can solve by PacBio, right?
Thank you so much. Yes.
Yeah,
hope so.
I actually fiber sick and SBB is really amazing. But actually, I saw in your presentation you don't have any comparison between PacBio and ONT. I saw it's just with Illumina, SBB with SBS, right?
Yep.
And for long read, you also compare with short read.
As well as
SBS
ONT as
is
well.
not with ONT. So do you have any research or result comparison between PacBio and ONT? I know that PacBio is perfect. I saw the PacBio result, but also now ONT they develop. There are many new versions. And actually, they also have. I think they develop the super accuracy for base calling.
Hmm.
And actually, they develop the flow cell. So quite good.
Yeah.
Yeah. They develop super accuracy the bioinformatic
Yeah.
tool, and the flow cell also is now is improved a lot. So do you have any data, the comparison between PacBio and ONT? So sometimes you said that PacBio give you the Q score 50, right? Even in the ctDNA, the liquid biopsy, and in the FFPE,
Correct.
you reach to Q50, but sometimes in ONT, I just read some information that they reach to Q20, 20 or 30 only, not 50. Cannot right now, but sometimes you don't need if because you have solution for any situation, right? For like liquid biopsy, cell-free DNA, and you have solution for the FFP. But sometimes it's just normal sample in blood sample. So now ONT also they develop for the whole genome sequencing for human. So do you have any suggestion or any comparison between PacBio and ONT result?
I
So
do have a lot, but I didn't put it here today, so I don't want to be like, keep bashing ONT, but I think the general statement from PacBio is always like, our accuracy is the best. We are the gold standard for long read sequencing, right? And like
yeah,
what you mentioned, right? For HiFi sequencing, our raw reads. So we didn't go through any base calling algorithm like AI. Our raw reads are already Q30.
yeah,
Don't need any base calling algorithm or AI or machine learning to actually help to modify the bases to achieve only, I think, if I remember correctly, it was like Q28 or Q29. So 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 that's one point. Like, our raw reads are already Q30. We don't need to go through any, like special base calling algorithm that keep changing every 2-3 months. Then you have to reanalyze the results so that you will have better base calling itself. So that is one point. And I think if you do um, if you do understand about like base calling algorithm as well, right? So we know machine learning is like you will feed like a set of data into it, they will learn through it, and then they will, look through, and then they will, modify, improve your base calling so that, they have the higher accuracy, but still not as accurate as us. So a lot of time, this kind of base calling algorithm as well, it will overcorrect, right? Maybe some of the mutations that are lower frequency, they will treat it as an error and then correct it. So you wouldn't. So that's a risk as well, right? So we know cancer genomics, you are looking at low allele frequency, like some of these somatic variants, right? So they might be present in the samples in maybe 5% or lower than 5% itself. So if you're using this kind of base calling correction algorithm, they might treat it as an error and correct it, right? Then you won't be able to really discover whether is it a real somatic variants or not. So I think that is another point that you have to uh, look at as well, lah. So for our HiFi sequencing, we don't do any base call correction. So every variance that we call, I think for clinical application, every variance matter, right? So that's why we feel that for PacBio, we still a lot of these clinical applications or diagnostic labs, they will still use our sequencer for do this kind of applications lah.
Thank you for your answer. And I've had last question. You mentioned about the genome polish, about the SBB. You mentioned about the gene genome polish. So what do you mean in that situation? Gene genome polish, because you said that you don't use the base calling, right? Software. So now HiFi PacBio, you just use AI to collect.
No, 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 no,
Do
no.
you mean?
No, I was saying our competitor.
Yeah.
Yeah, so I mean we have our own base callers as well lah, but our base callers are just to call from our HiFi sequencing itself. So there's no
Yeah.
correction algorithm in our base
Mm,
calling.
So what do you mean the genome polish? What do you mean in that case if you don't correct anything? So the meaning of polishment, the last I don't remember what the number of slide, but you mentioned about the gene polish in here. Where is it? In some of your slide, you mentioned about the genome polishment. Yeah. So what do you mean? Polishment, it means that some in some problem with base calling. So you need to correct it, right?
Cô nói là cái polishing á hả cô Diệp, tức là
Polishing, đúng rồi.
yeah, she's talking about like
Polishing.
the polishing.
Yeah. Genome polishing. I saw in your SBB slide.
Oh, did I? Wait, let me. Oh, why does it matter?
Yeah, genome polishing, yeah. Here. So in your red box.
Yeah, I see it.
So what do you mean genome polishing?
I can get back to you, but if I understand, maybe it's if you are doing like de novo assembly, like you want to make sure you have a complete genome itself. Sometimes it's short reads to
Yeah, right.
some of the polishing, right? And then I think maybe what they are trying to say, like for this kind of repetitive regions, sometimes you need to use this kind of like highly accurate to actually polish it so that you have a complete genome itself lah. But I can get back to you whether if it's
So do you mean that just only SBB short read, right? You need genome polishing, but for HiFi you don't need, because the accuracy is quite high. You reach to Q50.
Yeah, you are right. Like, for HiFi sequencing, because we are long and highly accurate, you don't need to actually do genome polishing you don't need to polish it with short read lah, to actually, achieve, probably to do like de novo assembly or things like that. Yeah, but I think for the short read side also, why does it matter here? It means probably, with highly accurate short read sequencing, it can be used for genome polishing. It will be able to enable better polishing. If let's say, you're using other long read platform, you need to do polishing, like with SVB, with this, like, because we can actually sequence through all this difficult to sequence region, right? And like this homopolymer region. Yeah, so it can be used for genome polishing. Yes.
Okay, so please, if you have any literature or some information about that, so please send me via email.
Yeah.
Interested in that, because it's very difficult for genome polishing. Yeah. So okay. So thank you, Raylene. Very nice presentation. Thank you.
Nice to see you again. I think I will work with Tue to provide you some of the literature.
Okay, thank you.
Thank you.
Cảm ơn các bạn, cảm ơn Thảo, cảm ơn.
Mọi người còn câu hỏi gì không ạ? Mọi người ơi. I think no more questions for you, Mr. Ray. Thank you once again for your joining us today and supporting Mitala as always.
No worries. Thank you so much for your time.
Thank
So
you.
if you all do have any, I mean, if the participants do have any question or, you want to work together with PacBio for cancer genomics research, do, reach out to me or DKSH to I think to is in the call as well. Yeah, to discuss more. Yes. Thank you so much.
Thank you. Thank you.
Thank you so much, Ray. Thank you for the team. Yeah.
Cảm ơn chị Tệ. Dạ vậy mình chào nhau cái mình kết thúc buổi hôm nay ha. Chào mọi người bật mic chào anh Ray đi cho anh vui.
So have a good evening, Ray. How are you over there? So what time in your country now?
It's 9:50. Going.
It's like the same, the
Yeah.
same, 9:50. It's not too late for you?
Yeah, time to sleep.
Yeah.
Thank you for your presentation.
Thank you. Thank you so much.
Today
Thank
is
you so much.
today weekend, you don't go out?
For Dr. Lloyd.
Okay.
Have a good night.
VIP person, right?
Yeah.
Okay. So thank you,
Bye
thank
everyone.
you.
Thanks a lot, Mr.
Bye.
Ray and chị Tuệ.
Thank you
Bye bye.
everyone.
Chào mọi người
Thanks
ạ.
everybody.
Thank Ray.
Give me
Bye.
Saturday. Thank you.
Cảm ơn mọi người.
Chị, bye bye chị.
Recording stopped.